For our next couple of topics, we're going to be hitting upon processes that rely on membrane transport quite a lot, both for thinking about salt balances and also for thinking about regulation and excretion of nitrogenous wastes. So this next week's lab is set up to help get us thinking a little bit more about how these processes actually work. And for doing this, we're going to have to start thinking about transport proteins and transport protein functions on two different sides of a cell's membrane. So we're going to start out studying this using an apparatus known as an Ussing chamber that's going to let us isolate a membrane and then distinguish between transporters that are found on one side of that membrane versus transporters and channel proteins that are found on the other side of that membrane. And I'll take you into the lab in a little bit to show you what the Ussing chamber setup looks like. But before we make any sense of that, the first thing I want to do is get you oriented to the tissue that we're going to be working with and a hypothesis about how carrier proteins and transport proteins are organized across this tissue. And then I want to show you something that will help you understand how we're going to be able to measure changes in transport processes across this tissue. So the tissue we're going to be focusing on is frog skin. So frogs engage in ion exchange with their surrounding environment across their skin. So when we're thinking about a frog skin, we're going to just designate two different sides to the skin's surface. One side, which we'll call the apical surface, is also referred to as the mucosal surface. And this is where the frog skin interacts with the surrounding environment. So we could also call this, where there's going to be pond water. Okay? Then the other side of the frog's skin, which is the interior portion, so it faces the frog's blood plasma, that side is referred to as the basolateral side of the tissue. It's also referred to as the serosal side. And again, that faces the blood. So as you'll see soon, for frogs, one of the challenges that they face is they risk losing sodium to the external environment. So they exist in an environment that has relatively low sodium concentrations. And in contrast, they want to maintain higher sodium concentrations inside of their blood or their bodily fluids because that higher sodium concentration is very important for regulating a lot of physiological processes. So now we need to look a little bit more at, okay, well, how do they actually maintain this across the skin? So they need to move sodium from an area of lower concentration to an area of higher concentration. And they actually do this with a set of transporters across each of these locations. I'm just going to show you what the hypothesis is, and then we're going to have a chance to test this hypothesis when we get to lab itself. So the hypothesis is that this gradient is maintained by the presence of a sodium-potassium pump on the serosal surface of these cells or of this membrane. Right, so this sodium-potassium pump is oriented such that it's going to move sodium ions from inside of these cells into the serosal region, right? So that's our net movement of sodium. And then meanwhile, this is our typical active transport pump, our sodium-potassium pump. So for a set ratio of sodium to potassium, for however many sodium come in, the appropriate amount of potassium then gets transported in the opposite direction, right? So this is an antiporter that requires ATP. Okay, so we've got that present along with that, then, the thinking is that on this serosal surface, there's also a set of potassium channels. Right, so this pump orientation means that there's going to be a buildup of potassium inside of this space inside of the cell. So then this potassium can actually just diffuse back down its concentration gradient through this potassium channel. So in some ways this is kind of a futile activity because potassium's pumped in and diffuses right back out. But what that means is that it's possible to then move sodium up its concentration gradient using energy. And then the next question is, well, okay, that's all well and good, but what about sodium from the external environment? How does that work? So thinking there is also that this is facilitated by the presence of sodium channels in the apical surface. Okay? Right, so collectively what this is going to allow is movement of sodium from a much lower concentration in the external environment into the cell. So at this membrane surface we've got facilitated diffusion, and then into this, or all the way into the blood plasma, we've got active transport at work. Collectively, this results in some electrical potentials at these two membrane surfaces. So at the apical surface, what this translates into is a membrane voltage here, we'll just call this VA, of negative 30 millivolts, right? So generally net positive charge outside of this membrane and a net negative charge inside of this membrane. And then on the basolateral surface, there is, again, a net negative charge inside of this membrane, a net positive charge outside of this membrane. So the difference here, which we can call VB, is, let's just say, something like 60 millivolts. And so then if we're trying to measure the voltage on either side of this, so let's say we have we're just going to draw a little voltmeter here, okay? And so this is going to be the transepithelial potential difference. So transepithelial, so that's all the way across this epithelial layer. So that's going to be, it's going to be our VB minus our VA, or negative 30 millivolts. Okay? So collectively, these transport processes are going to translate into a measurable electrical charge across this membrane, so long as these transporters are functional. So that's where the fun part comes in that part of what we're going to do is test this hypothesis about this arrangement of transporters across this tissue. And we're going to test this hypothesis by using a couple of different substances to kind of manipulate what's going on. So for testing this hypothesis, we have the following available to us. Typically, we're going to set up our chamber with a solution, a salt solution called Frog Ringer solution. And this has major types of cations and anions in it. So, you know, this is going to contain sodium, potassium, chloride, calcium, and a handful of other ingredients. In addition to that, we're also going to have potassium-free, or sorry, not potassium-free, sodium-free frog ringers solution. So we can add that to either side of our membrane, right? So here's our membrane. And so what I recommend doing is predict what will happen to transport, and therefore the voltage potential if the sodium-free ringers is added to one or the other sides. Right, so remember thinking here's our mucosal, here's our serosal side. Okay, so that's one set of tools. Along with that, we have a couple of drugs that have known functions. So one of the drugs is amiloride. And amiloride is often used to restore blood potassium levels back to normal in patients that have high blood pressure. And it does this by being a potassium channel blocker. So again, what I'd recommend doing is coming up with predictions based on that model of transport that I showed you for what you'd expect to happen if you added amiloride to the mucosal or serosal sides. And then along with that, we also have some wabane. And wabane is cardiac glycoside. And cardiac glycosides function by inhibiting ATP sodium-potassium pumps. And so again, I would recommend predicting what will happen if you add wabane to one or the other sides of this membrane. And collectively, based on those predictions, if your outcomes correspond with your predictions, that will help you determine whether or not this hypothesis about the arrangement of transport proteins is supported. At the heart of our labs this week are a set of chambers known as Ussing chambers, named after the inventor. And they're little acrylic chambers. They're small, but they've got a lot of things to them. So first off, they come apart, and you'll notice on the inside there's a little opening here, and there's a set of little prongs on the opposite side. So that little opening is where our tissue is going to wind up being put. So we're going to drape it across these little prongs. We'll also apply just a very fine layer of vacuum grease around the outside just to make sure we have a really good seal. Then everything will fit back together. And then what we'll do after that is there's two chambers here, and each of these will initially fill up with a saline solution, just frog ringer solution. So that contains a mixture of salts, and then it also has some glucose as an ATP source. Then once we've got that set up, to keep the tissue alive, it's important to provide an air supply to each side of this chamber. So there's a set of reduced tubes that look like this that will run into either side of the chamber. And that whole apparatus is then hooked up to a little splitter, and eventually that will run back to an air pump. So when you're ready to get started, you'll just plug that into the air pump, plug in your air pump, and then flow oxygen to both sides. You might also find it helpful to tape these tubes in place just so they don't wind up walking around. And in fact, you might find it easiest to just tape the whole chamber in place once it's ready. After you put it back together, just again to make sure that the connection between the two halves is very good, there's a set of these rings, clamp rings, and there's one set of spreader pliers which can be used to secure the clamp rings in place, just to ensure a very good seal for your chamber. Going to clamp that in. For the sake of the video, I'm going to skip that for right now, but clamp that in, then you are ready to go. Okay, so then what we're going to do next is we're going to set up a set of two circuits, a stimulating circuit and a recording circuit. So there's a wiring diagram for all of this on Canvas. I recommend having a copy of the wiring diagram handy because when you get to your table, you're just going to see a whole mess of wires, and you're going to be going, okay, well, what goes where? So I'm going to start actually at this connector. This is a BNC connector. Okay, so this BNC connector is going to connect up to the positive output here on the power lab unit. And that kind of twists on, so it's a slightly different connector type. From there, then, the next thing we're going to do is we'll start on the stimulating side, so I'm going to start on the red side here. If you look amongst your wires, you'll find a wire that has a banana plug at one end, and then it has a yellow connector at the other end. So you're just going to plug the banana plug in, and then for the yellow end, that's going to connect up to one of our stimulating electrodes here in yellow. So this just plugs together. For the actual lab, this is going to go into a glass capillary tube that has a salt solution in it, but for this demo right now, I'm just going to leave it as it is. This is then going to go into one half of your chamber, specifically onto the serosal side of the chamber. And inside of this chamber, there's a set of wells that run fairly deep. You can kind of see them on the side. So you can put your stimulating electrode there into the side or into a separate back well as well. So you just want to make sure that it's in there in a place where it's secure. It's not going to flop around too much. After that, then we're going to so at the other end of the stimulating circuit, you'll find another yellow connector piece. And this is going to connect into a piece of wiring that has a red connector. And the other end of it is an alligator clip. So this alligator clip has little jaws that open up. And then we're going to put a multimeter in line with this circuit. So here is my multimeter, and in this case we'll connect that red alligator clip to the positive terminal on the multimeter. There's a couple different ways you can make that connection. In this case, what I'm going to do is I'll just take the red tip of this multimeter and we'll make physical contact between these. If these alligator clips give you trouble, let me know. They're a little bit delicate in construction, so I might take a bit of work to get those connected correctly. Okay, then for the black end of my multimeter, I'm going to look for another alligator clip, double-ended alligator. And for this alligator clip, So I will clip one end of this to the multimeter again, and then the other end we're going to look for a black banana plug and a blue tip. We're going to connect up the other end of the alligator to the blue tip, and then we're going to run this black banana plug into the other part of the stimulator. And that's going to complete our stimulating circuit. So then the second circuit that we're going to need is our recording circuit. So for this circuit, we've got a homemade custom connector that interfaces with the inputs on the Power Lab. These are a little bit fragile, so please be careful while you're handling them so I don't have to solder things back together again. And for the recording circuit, so what I'll do is I'm going to just plug this into input channel 1. Okay. And then what I'll do is I'm going to take a cord here that has a yellow end and a blue end. Sorry, black end and a blue end. And with the black end, I'm going to plug the black end into a blue electrode, making sure that I line that electrode up on the correct side of the chamber. And then the other blue end will plug into the blue connection to the Power Lab. And then last but not least, I've also got a red recording electrode. And so I'm going to plug together the two parts of that red recording electrode, pop that into the opposite side, and then that's going to connect into the yellow connector on the input for the Power Lab. So that's going to get all of my wiring hooked up together. Alright, now the setup for this lab is a little more involved than the setup for other labs, so I've actually gone ahead and I've created a setup file for you. You'll find it in the labs module and with the file extension ADICHT, which is the file extension for lab chart. So you can go ahead and download that, and then if you load it, it will launch lab chart, and it's going to launch an interface that looks like what you see right here on the screen. Alright, so now I'm sure you're already noticing that things look really different here in this setup file compared to what you might be used to. The main thing we're going to be working with in this case is output in channel 1, and then we're also actually going to wind up looking at output on the multimeter. And in particular, when you're not using it, just try to keep the multimeter in the off position. But when we are in use, we're actually going to be measuring the current in microamps. So that's down here at the bottom on the multimeter. So I'm just going to set this down to the side for the moment. Also at the moment, instead of using the Ussing chamber, I'm just going to run some tests here with the solution in a beaker. So I do have a completed circuit right now. And this actually will show me a little bit of what the resistance is like across the entire circuit that I've created. Okay, so here in chart, we've got two different windows going now. The window right here that you see is the stimulator window, and then the other window is our usual kind of recording chart window. In this stimulator window, most things are set up for you. What we're going to work with is an option if you scroll down here, or if you enlarge this window. Whoops. So if you scroll down, you're going to see an option for adjusting the start height and the end height of your stimulus. And in this case, we want to apply a consistent stimulus, so we're going to adjust both of these measurements in parallel with each other. So to start out with, first thing you want to do, so get your multimeter turned on so you can record current. And then we can just start out running this. And if I just start out running this, you'll notice that my reading of my current is right now right around 0 millivolts. So what I'm going to do now is I'm going to step up this start height to 0.1 volts or 100 millivolts, and I'm going to also adjust the end height to 0.1 millivolts. And so you notice as I've done that, now my output voltage has changed across this circuit. This is small enough in this case that I'm not really seeing any change in my current yet, reading it so far. When you actually have skin in this circuit, because this skin should be generating its own current, your initial reading may not be at 0 millivolts. We want to get it to 0 millivolts. And it may or may not be the case that adding a current of, or a counter current, short circuit current of 10 millivolts counteracts the existing current in the skin. So you should see a change as you start to increment up the voltage, but it may take a little ways to reach your full short circuit current. So if I'm going to now go, I'm now going to go up to 0.2 for both of these. And again, you'll see this start to step up. And if you're, everything starts moving further away from 0 instead of getting closer to 0, what that probably means is some of your electrodes are backwards and you're going to have to switch them across the chambers instead. And then as you're doing this, you're going to wind up being able to detect a current with a multimeter. And there's a table at the end of the lab handout for recording both the stimulus voltage that you're applying and then also the resulting current in your circuit. So once you've got your wiring all configured and you've got the lab chart set up and running, and you're starting to record voltages and currents, once you've set your short circuit current, then it's time to start doing some experiments. So for the experiments, what that's going to involve is removing and adding different solutions into the Ussing chamber. And for the purpose of that, you'll have a set of syringes on your table that have some flexible tubing on the end of them. The biggest syringe here with the white tape on it is for discarding saline solution. So you just want to put that piece of tubing into each side of the chamber, draw up solution, and then discard into a discard beaker. And then depending on what solution you are adding, there are syringes that are color-coded for adding various different solutions across the different experiments. Notice, for doing that also, you're going to want to add specific volumes, so pay attention to the volumes that you're injecting with the syringes. And once you've finished manipulating the saline composition and concentration on your skin sample, then you're going to be adding amiloride and or wabane to either side of the chamber. So for that, you're just going to pipette in a small volume of the amiloride or the wabane, so just follow the directions that are in the lab handout. So hopefully this lab will give you a good sense of how physiologists study different tissue properties and figure out how different transport systems work across tissue types to figure out how organisms do things like bring in different ions if they need those ions, or excrete different ions, or move ions between different body compartments. A lot of it relies on just this simple technology, having a good sense of what's happening on each side of the membrane, and conducting simple manipulative experiments to determine where different transporter types are located. One thing I will say also for this lab is this is a lab where it's going to be really useful to spend some time and thought with the lab handout before you get to lab. So absolutely make sure that you have a good sense of what your predictions are across the different experiments. You're going to want to figure out someone to specialize on getting all of the wiring set up, and then you'll be able to have smooth sailing once you get to lab.